What's up, About Penguins? So today we're going to go over my responses to the 2025 FRQs. So the questions were just released yesterday, and the scoring guidelines have not been released. I am just an AP Bio teacher. I do not have any type of insider information. So what we are sharing are just my answers to the questions, and what I would have written. I'm probably going to fumble a little bit because I just looked at them, once to write my response. I haven't like, analyzed them to the point that I have them memorized. So our first question tells us that we have these cells, that there's certain proteins that are secreted and they're transported to the ER, either during translation or after translation. So the first question asks us to describe that function of the ribosome. So you think to yourself for a second, okay, what is the ribosome? What does it do? So the ribosome is responsible for protein synthesis. And so now it says describe. So I believe that we need to put a little bit more into it than just straight up protein synthesis, because that's more kind of identifying. So I figured that you need to describe it by, talking about how does it, what does it do really? And so there's two parts to the ribosome. The small subunit is going to bind to the mRNA, and the large subunit is going to bind to the tRNA. And so that allows for the mRNA to be read and the polypeptide to be synthesized. So I kind of gave a little bit more description to the process of that protein synthesis. So then they go on to talk about how there's this complex. And so based on having a signal, it's either going to move to the ER membrane to finish the translation, or it finishes the translation in the cytosol, and then it moves to that ER. So they talk about here that you need the protein SR for transport during translation, and then you need the SEC62 for transport after translation. And so if I was a student taking this, I would have like highlighted that, because that does seem to be important. They talk about that they're doing the investigating using different proteins, and they created this small interfering RNAs that reduce the expression of both the SR and the SEC62. And then they were looking at different groups of cells and kind of looking at how those proteins are all working together. So they gave us a figure. And one thing that I'm noticing is that I only have one standard error of the mean, which is kind of currently causing a lot of discussion in the AP kind of community about how they're going to take that. But let's just keep moving. And so they show us the graph showing our siRNA. So we have a control siRNA. So you can see that we have approximately the same amount of protein content in both of those, versus the SEC62 siRNA. We have a decrease of that one, but we have an increase of the the SR protein. So you can kind of see that the SEC62 siRNA decreased the amount of the protein that was synthesized because it interfered from that process. We also see that we have the SR siRNA, and the same thing happened here where I have a decrease in the SR protein because that RNA is interfering the synthesis of that protein. And so part B says to identify the dependent variable in the experiment. And so if you look at figure 1, you can kind of see the thing that the researcher is measuring is they're measuring that relative protein control content, and it's percent of the control. And so the dependent variable is relative protein content, as this is what the researcher was measuring. Then we have to justify why the researcher included the control of measuring the relative amounts of both the SEC62 and the SR proteins. So one of the big things about why would you use a control is for a basis of kind of comparison. So we're testing the control without the siRNA for those two different genes, because of the fact that we're trying to see, okay, what do the siRNA for the SEC6, I missed the number there, 662, and then the SR, how does the siRNA actually like inhibit those, okay? And so that allows us to compare to what would it normally look like, okay? And then number 3 or whatever, B3, based on the figure, describe the effect of the production of the SR protein when the cells are treated with the SEC62. And so if we look here at the SEC62, we see our SR protein, it does increase. And so here's normal right here, but we do see that it increases and the error bar does not overlap, which means that that data is statistically significant. So you say that there was an increase in the relative protein content. This is statistically significant as those error bars didn't overlap. And so that shows that that increase was significant. They then go on to talk about another experiment. Researcher measured the amount of each of 3 different proteins that was transported to the ER in the cells treated with the different siRNAs, and they calculated the percent transport. So we're seeing the percent of that protein that got transported there. And now that I've noticed it's talking about the measuring the amount of the different proteins, I'm kind of wondering if my answers are right in a second. So we're going to go with that. Identify the independent variable in the researcher's second experiment. So we're looking at independent variables. So what did the researcher change? And so the thing that's being changed is, of course, looking at the siRNA, which was the same thing that we saw in the first one. So my question then became like, were we looking at the different proteins? So I really don't know what they're looking for on this one. So I said the independent variable is the siRNA treatments that would be given to the cells. As I said, that is the same one that we saw at the first part, so I'm not really sure. So then I changed it to the independent variables are proteins 1, 2, and 3 under the control of the different siRNAs, because then we're able to really take into account the different variables that are taking to going on, but I'm not really sure. So then two, we wanted to, based on the figure, identify the protein at which the was treated with that showed the increase of the percent transport. Okay, so I did a little screenshot, and this is looking at just the control with the SEC62. That was all of my little diagram down here is going to show. So you can see here that it decreased. Those error bars do not overlap, so that data is statistically significant. Here I have an increase, and again, those error bars are not overlapping. You can kind of zoom in really close if you want to see the error bars do not overlap. And then here also, we're seeing these error bars are not overlapping, and it is increasing, so that data is statistically significant. So I said that protein 2 and 3 demonstrate a statistically significant difference from the control. Looking at that is going to be an increase of that percent transport. And so there's been a big argument about whether we're supposed to be looking at 2 SEMs. I think that you are fine to only look at 1, but if they were asking us to look at 2 and they wanted us to double it, then none of the proteins would demonstrate that significantly difference, because when you double this one, it's going to end up overlapping with that bottom. You can see that that spot right here is definitely going to overlap. And over here again, we're going to see that that overlap will take place. So I do believe that they're just going to look at the 1 SEM, not the 2 SEMs. So then 3, protein 1 encoded by 234 nucleotides, while protein 2 is encoded by 495 nucleotides. And then talk about how that this encodes all the nucleotides that are given will encode amino acids. That's important because the stop codon could have impacted this, as well as talking about like how that start codon. There was a question a couple years ago about how the start codon, that methane, not the methane, oh my God, methionine, sometimes kind of gets dropped off. So we want to calculate the difference. So we did the 495 divided by 3, because there's 3 nucleotides per amino acid, and the 234 divided by 3 again, because there's 3 nucleotides per amino acid, and that gave me 87. You could have also done 495 minus the 234 and then divided by 3 and gotten the exact same answer. So it really didn't matter which way you did this math. So then D, researcher claimed that protein 1 is the only tested protein that is transported to ER following its complete translation of the cytosol. Using the data from figure 2, we want to support the researcher's claim. So the SR is required for the transport of the ER prior to completing the translation, while the SEC62 is required for transport to the ER after completion. So if you look at the figure 2, you're going to see that protein 1 treated with the siRNA for SEC62 at the lowest amount of translation, denoting that it was inhibited from moving to the ER. So because of the fact that it's required to be able to transport to the ER after it was completed, and it didn't actually make it there, that's why we see there's a less amount of that protein. So then number 2, D2. For any protein that enters the ER, researchers claim that amino acids close to the protein's terminus determine how likely the protein is to pass through the protein channel within the ER membrane. Justify the claim based on your understanding of factors that affect transport proteins across membranes. So I'm thinking about how the membrane is a phospholipid bilayer, and how it has the hydrophilic heads on the outside and hydrophobic tails hydrophilic heads on the outside and hydrophobic tails on the inside. And so what my logic was is that those early amino acids must be nonpolar to allow for the beginning of that polypeptide to start making its way into that membrane. And so then once it's in the membrane, it may be able to then use those hydrophobic exteriors to then create some type of hydrophilic interior for it that we see like with our channel proteins. So I'm feeling like that there must be something about that membrane, about the protein that is nonpolar that allows it to get into the membrane and pass through the membrane to then allow for the formation of that protein in there. So that was the first question. The second question, many insects rely on pheromones, these chemical signals that are released by females to find mating partners. They hypothesize that a certain type of moth, the behavior is going to be regulated by a certain signaling molecule. So part A, the receptors are embedded in the plasma membrane. Describe the polarity of the portion of the receptor that is inside the membrane. I feel like this is very close to 1D, and I don't really know why. And so I feel like that one of my answers is probably not what they're looking for because of the fact that they're almost exact same answer. So the receptors inside the membrane is a protein. That protein has to be folded such that the hydrophobic R groups are facing outside. So they're going to face toward the hydrophobic fatty acid tail. And then that's an extracellular portion that will then bind. So then so we've got this part that's sitting in the membrane, and then there's going to be a little portion that's going to be outside of the membrane that's going to be hydrophilic. And that way it can interact with the aqueous solution, and then it will be able to receive that signaling molecule. So to investigate the binding of that receptor of a certain DOP ECR, they then look at kind of inhibiting it with siRNA again. And so then they're going to investigate kind of the general activity of it, and then how much of that general movement is oriented activity. So they're looking at the different treatments of a control, which just had saline, versus some that have the siRNA. And so we're looking at the percent of that total time. So 95% of the time with the control solution, they had general activity. And of that 95%, 60% of the 95 is what we see with oriented activity. And then we see where there's 90% when we gave it the siRNA, and then 25% of the 90 is what we see going on with the oriented activity. And so when they bind to it, we see that GTP displaces the GDP. And so you see that activation pathway where it's then going to synthesize the genes that are needed for that oriented activity. Okay. So we want to use the template to provide for the response. So I asked after the questions were released, what did the graph look like? And so my students told me that the graph paper was kind of narrow and tall. So I do not know what your graph paper looks like. As you saw when they released the questions, they did not release what the graph paper looks like. So you were the only ones who saw your graph paper. So this is just my estimation of what the graph paper looked like. So your graph could look slightly different because your graph paper might have been different than mine. But so on our y-axis, we're going to put the activity. So I just did percent, and then the average plus or minus the standard error of the mean. And it did give us 2 standard errors of the mean in our data table, as you see here. So we do have the 2 that we're then going to use for our data. Okay. I did mine where each of my blocks were worth 10. I mean, sorry, we're worth 5. So that we would easily be able to see and use as much of the graph as possible. So we plotted our points at 95. 1 second, let me make it smaller so you can see it. 95, then we have our 90. Then we have our 60, and then we have our 25. And this is the 60 right here. So I was looking at the control. What did I do? Oh, I looked at the control, and then I was looking at the inhibited one, because my logic was I wanted to kind of put the controls together and then the siRNAs together, so I can kind of see kind of overall what was happening between the general and the oriented, since this is a fraction of that. At least that was my attempt to do. So I think that the points for this are going to be that you appropriately labeled your axes, labeled it with units, and that it's scaled correctly, that the data is appropriately plotted as a bar graph, and then there's error bars appropriately plotted. I cannot see this as any other graph because I don't have any type of linear data, and it's not like individualized data, so it's not going to be a scatter plot. So I do believe that this is a bar graph that we're going to see here. So then we're going to determine the type of activity that's affected by inhibiting the expression. So by inhibiting the expression, you're seeing that the oriented activity was decreases because of the fact that we have this lowest amount of the siRNA treatment from that oriented activity. So you can kind of see that it's a very low percent there. So I think it was the oriented activity that was inhibited. Okay. So then based on the table, identify the treatment group in which the orient activity was greater than 50% of the general activity. So we're looking at that the control group had 60% of the what's called, so that was 60 plus or minus the 4, still gets us greater than 50%. So I said that it was the control group injected with the saline. So then scientists studied some moths with mutation in the gene encoding the G protein that prevents the GTP from replacing the GDP. So predict the effect of that mutation. So if I don't have this step taking place here, right? So the GDP can't be replaced by the GTP, then this entire pathway isn't going to take place. So we're not going to activate the G protein, and we're not going to see that our genes are going to be increase their expression. So we're probably not going to see the oriented activity taking place in those moths. So based on the diagram, it's not replaced, and then that means that the G protein is not activated, so there will be an increase in the expression. So then there's a decrease in the oriented activity of that male moth. So they talk about how the expression is low in the males during the first few days of adults when they're sexually immature, so they can't reproduce. The gene expression then increases as the moths reach sexual maturity. So the scientist claims that the increase is due to them finding a female with whom to mate. So use evidence to support. So the pheromones are designed to attract a mate. So if the males are sexually immature, then they're not going to be suitable from like for the first few days as adults. So there'd be a lack in that gene expression. Once they reach that sexual maturity, we're going to see the increase of it in order to respond to the signal from that nearby female to mate. They probably want a little bit more for it. I'm not really sure what the AP scoring guidelines are going to look for. So then explain how the inhibitor might serve as an effective chemical to protect the crops. So I think that moths reproduce on crops. And so if I am trying to protect my crops from the damage from the moths, then maybe the fact that they're laying their eggs in the crop is damaging them. So my thought was that if we can decrease their oriented behavior, decrease them responding to the pheromones, then we're going to have a decrease in the crop kind of damage. But I feel like that also just decreases the reproduction of the moth. So that kind of makes me think that that might not be good for the environment if we're decreasing a certain organism's ability to reproduce. But I'm not really sure where they were going. So then 3, apparently this is your favorite question: the buffalo grass and the invasive species. As threatens as cactus, it's a ketone species. They conducted an experiment in which they controlled the abundance of it and identified different native grass species. And so they did it where they put it under non-drought conditions where they watered every 3 days, and then they put it in a drought condition where it's watered every 9 days. And then they measure the height and the dry weight of it. So the effect of a ketone species, this is a generic answer because every time you remove the ketone species, it has a drastic impact on the ecosystem. It's going to cause a collapse because it changes those population dynamics, which decreases the biodiversity, which then decreases also the resiliency of that ecosystem. So I'm not really sure where in all that is where the answer is, but that's kind of what's happening. A control group of this is going to be one that doesn't have the the native species. Since we're specifically testing, the scientists identified several ones. There was something about how they were testing those different native species. And so since they're testing the native species, they want to see, oh, wait, here it is. They identified several native species where grown might reduce the abundance of the buffalo grass. So since they're trying to determine which of those are going to kind of reduce the abundance of it, we want to test the native species to be able to look at that. It gives us a baseline, so we'll be able to then compare the different conditions with the buffalo grass, okay? So then we want to state the null hypothesis, and as I've always said, this is the same way I do it always. The presence of the native grass is going to have no effect on the abundance of the buffalo grass species. So independent variable has no effect on dependent variable. Thanks. So then the scientists are going to then found that the population growth rates are slower than the buffalo grass when we have a wildfire. So the logic is like, they claim that there's an increase in the abundance of it. So there's kind of 2 thoughts that went through my mind. First, that we have this wildfire. The wildfire, of course, has already said that there's a higher population growth rate than the native grasses. So since it's growing at a faster rate, like its slope is going to be higher because it's got that higher rate. It's going to have more births and deaths and all that fun stuff. But then my thought also is that if you look at the original prompt, it talked about how the buffalo grass can survive these wildfires. So if the starting amount of it is a larger amount, then it's going to grow its abundance a lot quicker than if it started a lower amount. So that was also my thought process, is that the fire is going to reduce the population size of those native grasses, but it doesn't reduce the population of the buffalo grasses. So they're already at kind of an advantage to grow and become faster, kind of more abundant. So then we have the 20 million years ago, we've got the Caribbean Sea and the Pacific Ocean. They were originally connected. We have free-flowing water between those two. Then the Panama kind of came and closed that connection between them. And so we no longer saw this movement of the water. And then they talk about like one of them is nutrient rich, and then one of them is it's warmer. So described evidence, genetic evidence that evolution occurred. So looking at this, we're talking about how if you think back to like Hardy Weinberg, we always talk about that if the allele frequency change, if the genotypic frequency change, that means evolution is occurring. So if we're looking for genetic evidence, the evidence is really like the allele frequencies or the genotypic frequencies, and if those change. B talks about explaining how the isolation of the barrier or isolation of the species due to the barrier can lead to divergent evolution. And so this has to do with speciation. We have allopatric speciation taking place. There's a lack of gene flow. So we have this reproductive barrier of a geographical barrier, if you may. And because of the fact that these populations are now separated, we could have where there is a mutation that happens in this population that doesn't happen over here. And as this continues to accumulate over time, we'll see that the two populations will diverge and become two different species that if they were to come back in contact with each other, they may not be able to reproduce, which then of course makes them different species by the biological species concept. So then they talk about the formation of it, connected the two continents. So we have North America and South America that are now connected to each other. And so the North American animals then migrated south to South America, and they came to similar niches as the South American species. So we want to think about the effect of that on the resource availability. So if there are all these resources down here in South America, and now the North American organisms have come down, they're going to use some of those resources. So we're going to see a decrease in the resources, as well as they're using similar niches. So we're going to see competition taking place to the point that maybe we might have resource partitioning, or we're going to lose certain species because of the fact that they're not going to be able to obtain enough resources for themselves. So then 5, figure 1 shows reaction of the metabolic pathway using the synthesis of amino acid B from amino acid A in the cell. So you can see that we have amino acid A right here. It's going to bind to enzyme 1 that then allows it to make the product of intermediate X that binds to enzyme 2 that makes the intermediate of Y. Y binds to enzyme 3. And then we have our final product of amino acid B. We can see that B is going to come back here and acts as a non-competitive inhibitor, and then it kind of to inhibit the synthesis of amino acid B. So we see like some negative feedback taking place. So then A, describe a characteristic of enzyme active site that allows it to catalyze a specific chemical reaction. So my thought is that it has the active site, and that active site is going to be specific for binding, so we can bind a specific substrate to it. And when it binds it, it can also strain those bonds. It can also kind of decrease the activation energy, which is what the function of an enzyme is to decrease activation energy, which increases the reaction rate. And so I'm kind of thinking that between the bonding of it, it's kind of specific to the size of the substrate and the charge of the substrate and those specific groups, as well as the decreasing of the activation kind of energy is where we're looking. Explain how the binding of amino acid A to enzyme 1 is regulated by amino acid B. So as we already talked about, B is going to come back here, and it's going to bind to enzyme 1, acting as a non-competitive inhibitor. By binding there, it causes a conformational shape change. And so now amino acid A is no longer going to be able to bind to enzyme 1. And so we're going to see negative feedback taking place. So amino acid B acts as a non-competitive inhibitor, binding to allosteric site, changing the shape, and then that's going to decrease binding of A to enzyme 1, which then inhibits the synthesis of the B. So we see our negative feedback. C, use the information in figure 1, identify the product of the reaction catalyzed by the enzyme 2. And so here if we look at enzyme 2 starts out with intermediate X as its substrate, and then it synthesizes intermediate Y. Y continues on and uses enzyme 3, so it becomes a substrate of enzyme 3 to make amino acid B. And so what we're looking at is what is the product of enzyme 2? The product of enzyme 2 is intermediate Y. And so this was different than I was expecting, because usually we have 5 being some type of modifying the diagram, but this one they just had you identifying a product, which was kind of cool. So it's just Y. And then I'm just explaining why it can't be the other ones. D, based on figure 1, explain how a change in pH could affect enzyme 3 in such a way that amino acid B cannot be produced. So here we see that enzyme 3 is kind of what is catalyzing the reaction from intermediate Y to amino acid B. And so if we have a changing of the pH, that could denature enzyme 3, and so it's no longer being able to speed up that reaction from intermediate Y to synthesize the amino acid B. And so we're going to see that denaturation could then decrease that reaction rate. So then we move on to question 6. So we have this all gene of fruit flies. It encodes a certain protein that's associated with both of the centromeres of chromosomes and protein filaments produced during meiosis. In the absence of it, we're going to see that it goes into phase 1 because before they're actually aligned. And so we go through process of meiosis a little too soon. So there are 4 different mutations made. We have ALT1, ALT3, and ALT23, and then the del. The del involves the deletion of a gene. So then we're seeing that we have all these different genotypes of our organisms. We have a wild type. Then we've got the wild type with the deletion, the ALT1 with the deletion, the ALT3 and the ALT23, and then the ALT23 and a deletion. So we're kind of seeing 3 of them that have deletions, and then the other ones are just modifications of mutations. So here we're looking at the percent of cells that are in metaphase. Metaphase cells with the different filaments. And so you can kind of see the wild type is here. Then you've got the wild type deletion, the ALD1 del, and the del3, the ALT3 and the ALT23, and then the ALT23 del. So you can kind of see that we deleted the gene, and then this one apparently is just not there because we have absolutely no cells, and we're really seeing a rapid decrease going on there. And then they give us the amount of protein. So I'm assuming this is some type of gelatiophoresis. It does tell us up here, the thicker band indicates a rate or amount of that protein. So we can know that there's most protein is made in the wild type, with the least amount of protein is in the ALT23 and the del. So part A, based on the figure, identify the fly genotype in which the average percent of metaphase cells is close to 12%. So when I did the little screenshot and I was looking at the bottom, I can see that this one is about 12%. And so that would be the ALD1 del is where we have the closest to 12%. Then B says, based on figure 1B, describe the difference in the ALD protein production between the gamete-forming metaphase cells with ALD3, ALD23, and then the ones that have ALD23 and then the del. So you can hear we have more of the protein synthesized in the ALD323 than is ALD23 del. As you notice, there's actually really nothing there. And so, of course, we see that the ALD323 is higher than the ALD23 deletion. So then C, they hypothesized the gamete-forming metaphase cells can produce a normal amount of the filament even when they produce about half as much ALD protein. So we want to use the data to support that hypothesis. So here you can kind of see that we have about half as much protein synthesized when we have the wild type del versus the double wild type. And so if we look here at the graph, we can see that the error bars overlap. So though the wild type del kind of looks like it's higher, they actually have the same percent of cells together. So even though this one has less of the protein, they have the same amount of the percent metaphase cells. And so the logic that we have here is that they're statistically the same, and that there must be something about that it produces still the same amount of the filaments even though it has half the amount of the protein. But our data that we support is pretty much the graph. And then D, for gamete-forming metaphase cells of the wild type del and the ALD1 del, explain why the phenotypes observed figure 1 differ even though the amount of the protein does not. So here we see we have the same amount of protein, approximately. They're both about the same width in terms of the amount of protein. But if you look at the graph, we have a substantially lower amount here with the ALD1 del. So my logic is that if we have the del, so here we go from wild type to del, we decrease the amount of protein in half. So the wild type makes, let's say, half of the protein. So we have half the protein made from wild type, half protein made from the other wild type, which gives us the full amount here. This one I have wild type, so half, and then the deletion, of course, was deleted, so we're not making that second amount of the protein. And then here I have the ALD1 in the deletion. Again, I'm able to make protein, and the second one is deleted. So we still have a gene that's making protein. But the difference is here is that maybe that mutation that took place in ALD1 leads to a non-functional protein, or maybe the protein is not formed the same way and it kind of has a different function. And so what I'm thinking is that that mutation leads to a difference in the ALD associated filaments, which then leads to a lower percent of the metaphase cells. And so I think I talked through that, comparing those, looking at that. I think that one synthesizes the other. So we have the wild type to ALD change represents that gene is still present, and the mutation could lead to a difference in the function. So I hope that this was helpful. You can ask any questions you want in the chat, or not in the chat, but into the comment section. I probably will not have answers for most of your questions because of the fact that the scoring guidelines have not been released. I, again, as I said, have no insider information to this. These are just what I came up with as I try to put them together this morning at 2:30 this morning. I don't know, whatever. But you're welcome to ask any questions. And as always, AP Bio just to satisfy y'all.